Hello, I'm Elena Klimova and I'm from Ukraine. I want to present you my scientific project method of bacterial cellulose production from plant waste substrates. Nowadays, one of the most concerning environmental issues is polyethylene pollution, caused mainly by a long period of polyethylene decomposition. The main alternative to polyethylene is paper, but the increased use of wood as the main raw material for pulp production leads to intensive deforestation. The most profitable alternative to traditional way of receiving cellulose is its microbiological synthesis. The ability of some bacteria species to produce cellulose has been known for a long time. However, the main problem of its industrial production is insufficient productivity of the process. That is why my goal is to develop a cheaper and more effective way to obtain this material. Before I started my researches, I had studied the morphology of producer, the Mrs. gisevi, with an addition of cellulose-producing bacteria strains, Gluconobacter oxidans and Gluconocetobacter hansenii. The culture of producer was plated on modified carb medium. This is an indicator medium, therefore, with the growth of producer's cultures, yellow zones appeared around its colonies. They were examined by light microscopy at different magnifications. Thus, I discovered that the membrane contains cellulose-producing bacteria species, namely Gluconocetobacter xylinus, Gluconobacter oxidans, and Gluconocetobacter hansenii. Strain combination is author's development. At the next step, I developed various types of organic substrates made of organic waste, group 1, organic waste with an addition of acetic acid, group 2, ethanol, group 3, vegetable oil, group 4, and meat molasses, group 5. Control group is a sucrose solution with concentration of 54 grams per liter. Organic waste, fruit and vegetable peels, was washed with running water and grinded for further homogenization of the culture medium. To this mixture, 1 gram of cellulose was added, and then it was heated to a temperature of 60 to 65 degrees Celsius. The temperature was maintained for 40 minutes. After boiling, the mixture was filtered and sterilized by the ultraviolet sterilizing lamp. Then distilled water in ratio of 1 to 3 was added. Culture medium preparation for other groups was specified by the addition of secondary components. For the second group, 4 milliliters of glacial acetic acid per liter. For the third group, 10 milliliters of ethyl alcohol per liter. For the fourth group, a few drops of vegetable oil. Fifth group is a beet molasses solution in ratio of 1 to 3. All the substrates with inoculated with the Mrs. gisevi cultures was placed in air-dry incubator with a temperature of 28 degrees Celsius. The cultures on the substrates were grown for 72 hours. Then the cellulose films were removed and weighted. I concluded that the most productive culture mediums were group 2 substrate, organic waste with glacial acetic acid, and group 5 substrate, beet molasses. There is an average about 36 to 38 grams per liter of cellulose synthesized on these substrates in 72 hours. The obtained data was analyzed using students' t-test. Physical properties of obtained material were demonstrated with the help of self-made bag. 2 pieces of cellulose film were glued with sucrose solution. Considering the cost of the raw material, less than 1 cent for 1 ton, the cost of a ton of bacterial cellulose would not exceed $30. At the same time, market price of a ton of wood pulp can be higher than $940. To determine the time and optimal conditions for the cellulose film decomposition, a small piece of this material was placed in a container with moist soil at a temperature of 25 degrees Celsius. A similar piece was left on the surface. The condition of the test sample is regularly checked since the 7th of November 2020. Therefore, the bacterial cellulose period of decomposition was determined, 2.5 months on average. Thus, with the developed technology, the cost of bacterial cellulose has been notably reduced by a cheap culture medium and increased productivity of cellulose synthesis. Thank you so much for your attention. I hope that my project will help to change the world for the better.